In this video, we will discuss a workflow and techniques for isolating single cells infected with a chytrid, establishing a dual host parasite culture, and sequencing your isolates. This video builds off our previous one on sample collection, which is linked to in the description below. We will first discuss how you can isolate single chytrid cells from a water sample using a capillary mouth pipette, followed by ribosomal DNA and whole genome sequencing. Another common single-cell isolation method not discussed in this video is fluorescence-activated cell sorting. Following that, we will discuss how you can establish a dual host parasite culture and a method for sequencing the community. Isolating single infected cells with a capillary mouth pipette and single-cell sequencing. Here, Dr. Cito explores a water sample for infected cells. For example, here is an image of a diatom infected with a chytrid. As an expert mycologist and phycologist, Dr. Cito can spot even the smallest infected algae, like this alga infected with an aphid. Once a chytrid of interest has been spotted, single infected hosts can be isolated with a glass microcapillary mouth pipette. Dr. Cito takes a sterile glass pipette and holds the end over a flame. After a few seconds, the glass is hot enough to be stretched. With one quick, precise movement, the glass is pulled into a long capillary and the end trimmed in the fire. Dr. Cito makes this maneuver look easy, but it can be pretty tricky, so try to be patient when you give it a shot. Next, Dr. Cito uses forceps to break off the end of the capillary at the desired length by applying pressure, wiggling slightly, and pulling away. Dr. Cito then adds drops of autoclaved and UV radiated deionized water with a P1000 pipette to the cover of a sterile petri dish. The chytrid sample will be cleaned by serially transferring it from one drop to the next. Dr. Cito uses his mouth pipette to suck up the sample, in this case, a single infected diatom. Under the microscope, the sample is expelled into the first drop. It is helpful to always keep your eye on the sample by focusing on the tip of the pipette as the sample is expelled. Dr. Cito transfers the sample from one drop to another, removing as much debris as possible and diluting any contaminant bacteria. Here is an image of the final rinsed cell. Finally, the sample is placed in a 0.2 milliliter PCR tube. The single chytrid cell can then be processed for DNA sequencing. After first extracting the DNA, the genome is amplified with techniques such as multiple displacement amplification or degenerate oligonucleotide PCR. This results in larger quantities of DNA required for sequencing. The entire ribosomal DNA operon can then be amplified with primers developed by Wurzbacher et al. 2018 and sequenced with Oxford nanopore. The sequencing of both highly conserved and quickly evolving regions of the rDNA operon allows for robust phylogenetic placement. In addition, the genome of the single cell can be sequenced by fragmentation, library preparation, and Illumina sequencing. With the genome in hand, different bioinformatics analyses can reveal much about the ecology and evolution of the organism. Alternatively, the single cell can be used to help establish a dual host parasite culture, which we will talk about next. Establishing dual host parasite culture. Here, Dr. McKindles uses a handheld pipette man to isolate algae for culturing. The target organism is transferred to liquid media. For example, WC is a common medium used for growing freshwater diatoms, but you will need to select the appropriate medium based on the needs of your target organism. By repeatedly transferring the target cell to fresh media and excluding as many other organisms as possible, Dr. McKinnell can dilute out contaminants and eventually establish a unialgal culture. This same method can be used to isolate cells parasitized with chytrids. Following the establishment of a host parasite culture, total DNA from the simple community can be extracted, prepared for Illumina sequencing, and sequenced. Bioinformatics pipelines can then be used to assemble and differentiate the genomes of the host and the parasite. In summary, we discussed methods of isolating single cells infected with chytrids using a mouth capillary pipette method and serial dilution, the establishment of a dual host parasite culture, and sequencing single cells and host parasite communities. That's it for this video. Thanks for tuning in, and good luck finding chytrids.